Hello all, today we are going to learn about nitrogen metabolism. In this nitrogen metabolism, we are going to see about nitrate assimilation, ammonium assimilation, nitrogen fixation, root nodule zones, regulation of nitrate reductase, photorespiration and nitrogen recycling, amino acid families. Let us start from the nitrogen metabolism overview. Plants cannot take nitrogen directly from the atmosphere. Atmosphere has approx 78% of nitrogen. Even though atmosphere has 78% of nitrogen, plants cannot take them directly. So they can take them as ammonia or nitrate. So these nitrogen have to be converted to ammonia or nitrate by using mechanisms. In these mechanisms, there are several enzymes involved. Let us see them later, okay? First, let us see why this nitrate or this ammonia is required. This is required for producing amino acids which are not available in plants, okay? These amino acids converted to proteins later. Here you can see in the diagram simply, atmosphere has 78% of nitrogen. Here nitrogenase enzyme is involved. So we need lot of energy, that is 16 ATP is required. Also it is combined with 8 protons and 8 electrons. So it formed 2 molecules of ammonia and also hydrogen gas. Okay. In legume plants there is Rhizobium bacterium is involved. So this bacteria does root nodules. So it converts atmospheric nitrogen to ammonia or ammonium. Okay. There is some pathways involved in this ammonium metabolism. So let us see them later. Okay. Let's go to next slide. Nitrate assimilation means conversion of nitrate to ammonia. Okay. It occurs with 2 enzymatic activities. They are nitrate reductase and nitrite reductase. Let us see how it happens. Nitrate plus NADH. NADH or NADPH is utilized here. So it forms NO2. That means nitrous oxide, and also forms H2O. That means water. Okay. It happens in cytosol. This process happens by using light. Okay. So light is required for this process. In the first step, you can see that nitrite is formed. NO2 minus. Okay. It reacts with 6-ferridoxine and forms NH4. That means ammonium. So it formed ammonium ions. Okay. It happens in chloroplast and also in plastids. Plastids are present in roots. We can see what are plastids later. Okay. So the first one is happening in leaves. I explain you simply again. So what happened here? NO3 minus, that is nitrate, converted to nitrite, that converted to ammonium. Okay. This NO2 is toxic. This ammonia also toxic. Okay. Here you can see that in diagram. GS-GOGAT pathway. GS means glutamine synthetase. Okay. So it synthesizes glutamine. We know ammonia was formed here. So it reacts with initial amino acids, that is glutamate, which is present earlier. This glutamate reacts with ammonium using some energy, that has ATP, and forms glutamine. These reactions happen in cytosol as well as in chloroplast. Please remember this all. So what happened previously? Glutamate is merged with NH4. That means ammonium. So it formed glutamine. Now this glutamine reacts with alpha ketoglutarate, which is present earlier. Alpha ketoglutarate. Okay. It forms 2 glutamate ions. So what happens now? These 2 ions again forms glutamines. 2 glutamines later. So what are the electron donors here? NADH. Please remember it. And also ferridoxine. Please remember it also. But what is the purpose of this glutamine or glutamate? Why it is required? Why it has to be synthesized? Do you know? Let us see why it is required. It is a central amino donor molecule. So it is required for everything. So this glutamine is required for next steps. So this has to be synthesized. So this GS-GOGAT pathway happens in plants. Let's go to next slide. Ammonium assimilation in plants. We have seen it in earlier slide. So that applies here. Glutamate that merges with ammonium using energy and forms glutamine. So we have seen earlier. So this is ammonium assimilation. Plants cannot utilize atmospheric nitrogen. But why? Because nitrogen has triple bonds. Due to triple bonds, it is more stable. So it cannot be broken. So it has to be converted to ammonia or ammonium, okay? So that only plants can absorb it. In legume plants, there are some microorganisms involved for doing these actions. The first one is symbiotic microorganism, that is Rhizobium. There are other microorganisms also, that is free living microorganism, Azotobacter, Clostridium. Okay, do you remember Clostridium botulism? It causes food poison. Okay, the another microorganism is associative. It lives with plants. Okay, that is Azospirillum. What are legume plants? They are peas, beans, groundnut, soya bean, chickpea. Okay, so this nitrogen fixation happens in these legume plants. Now let us see about how root nodules occurs. That means how nitrogen fixation occurs in plants. The first step happens with chemical signaling. That means plant releases some flavonoids. Okay, these flavonoids attracted by Rhizobium bacterium. So it releases some chemical compounds. Let us see what are the chemical compounds it releases. It releases lipoketo oligosaccharides. So these molecules acts as a signaling molecules to the plant. Okay, by these signals root nodules will be formed. We call it as shepherd crooks. These infected threads like tubules. They are like tubules. I cannot draw it perfectly, but they are like a tubules. Okay, this nitrogen fixation happens by nitrogenase enzyme, which is produced by plant. Okay, nitrogen reacting with 8 protons, 8 electrons and forms ammonia and hydrogen gas. Okay, we have seen it earlier, right? Let's go to next slide. Structure of root nodule zones. It is a 5-zone model. Okay, I forgot to tell you one thing, that the Rhizobium requires oxygen, but nitrogenase enzyme won't work if oxygen is present. So how plant solved this problem? It's solved by leg hemoglobin protein. It binds with oxygen but maintains low concentration of oxygen in the plant. So nitrogenase won't affect by it, but Rhizobium gets oxygen. So it's solved by leg hemoglobin protein. Please remember it. Let's come to the point. In this root nodule, okay, there are 5 zones. Let us discuss about them. The first one is meristematic zone. It is present at tip of the nodule. Okay, so it is at end part. Okay, so here is no bacteria, but here is actively dividing cells of plants. It is responsible for nodule growth. The second one is infection zone. So bacteria is present here. Bacteria moves through infection threads. Plant cells enlarges also, and bacteria enter the cells and starts differentiating. So here Rhizobium is mixed with root nodule. So we call it as symbiosome. Zone 3, that is nitrogen fixation zone. You can see that the color is changed to pink. Why it is in pink color? Because leg hemoglobin in the pink color. We can see here it is in pink color. It is important zone for fixing nitrogen, right? Here bacteria differentiate into bacterioids. Also nitrogen fixation happens actively. Here leg hemoglobin concentration is very high. That's why it is in pink color. Here N2 converts into ammonia by using nitrogenase enzyme. There is one zone is missing in the diagram. Let us see about it. The fourth zone is senescence zone. So here opposite reactions happen. Here nitrogen fixation reduces. Also bacteria that are bacterioids declines, means degrades. Okay, so nitrogen fixation is reducing here. That's why we call it as senescence zone. Okay, the fifth step happens in saprophytic zone. So here we have seen that nitrogen fixation was reduced and also bacterial cells are degraded. So now plant cells will die, and also the remaining bacteria releases into the soil, and also nutrients recycle happens here. So nutrients releases into the soil. Okay, so root nodulations are stopped here. There is so much to explain, but I will explain it in next class. Please stay tuned. Thank you for watching.